So first, I'd like to thank ADAA for inviting me to give this talk, and to those of you in the virtual audience for taking the time out of your busy schedules to come and hear about this work. I feel very honored and very excited to be here. So today I'll be discussing a particular research project of mine, which was selected as the best original research paper and resulted in my receipt of the 2022 Donald F. Klein Early Career Investigator Award. This paper is now published in the Journal of Depression and Anxiety. So if I pique your interest enough today to go and give it a gander, you can find it there. So I'd like to start by very briefly introducing myself. I'll then provide a review of some of the relevant literature that informed the work I'll be discussing today. Then we'll get into the real bread and butter of this talk, where I'll discuss the Donald Klein Research Project. And lastly, I'll leave a moment at the end to talk about my experiences as the ADAA Donald Klein awardee, and also to provide contact information. Since this is a pre-recorded talk, I welcome you to contact me with any questions or comments that you might have. So just a little bit about me. I received my PhD in clinical psychology from Virginia Commonwealth University, where I trained with Dr. Ananda Amstatter in her lab at the Virginia Institute for Psychiatric and Behavioral Genetics. And this setup really allowed me to have a unique and blended training experience, where I had the clinical training from VCU's wonderful clinical psych program, as well as the statistical and behavioral genetics training through the PhD program housed at VIPBG. I obtained an F31 award from NIAAA, which provided me this supplementary training that I really felt I needed for this career that I knew I wanted, better understanding biological processes that underlie trauma-related conditions and comorbidities. I completed my clinical internship at VA Puget Sound in Seattle, where I worked with Dr. Tracy Simpson. And there I really expanded my understanding of PTSD comorbidity more broadly, gained exposure to treatment outcomes research, and the use of large epidemiological data sets. Fellowship then brought me back to the East Coast, where I'm currently wrapping up my second year of T32 funded postdoctoral training at the National Center for PTSD and Boston University School of Medicine. There I work with Dr. Erica Wolf and her team. And I intentionally sought out a fellowship where I would get the training necessary to expand this foundation in behavioral genetics, to include approaches that would enable me to investigate downstream health effects of trauma-related comorbidity, with the long-term goal of informing behavioral and precision medicine efforts. This will really be the primary focus of my talk today. And in a few short months, I will be starting a faculty position at Old Dominion University in Virginia, where I'm very excited to continue expanding my line of work. Okay, so let's get into it. Trauma is highly prevalent. Most people experience trauma at some point in their lives, with the majority of individuals experiencing multiple traumatic events. Trauma is highly heterogeneous in that it can take many forms. And although different trauma types are associated with varying degrees of conditional risk for trauma-related outcomes, so for instance, trauma that is interpersonal in nature is associated with increased risk for PTSD, for example. But trauma exposure broadly, it increases risk for a number of mental, physical, and psychosocial health outcomes. These are just a few of many examples listed here on the slide. This really emphasizes the fact that trauma is a transdiagnostic risk factor for both psychiatric and physical health outcomes, and that better understanding the processes by which trauma affects the mind and the body has important implications for the majority of the population. PTSD is unique from other psychiatric disorders in that it's often regarded the signature disorder of trauma exposure because exposure to a traumatic event is a diagnostic criterion for PTSD. Nationally representative surveys suggest that upwards of 9% of individuals will meet PTSD criteria for PTSD at some point in their lives, with the highest conditional risk being accounted for by those having experienced multiple traumatic events. Research also shows that having a prior history of trauma increases one's exposure to subsequent traumas, further exacerbating risk for greater cumulative PTSD or PTSD symptom severity. Meta-analytic studies have found strong evidence that having a PTSD diagnosis increases one's risk for chronic disease, including but not limited to cardiovascular disease, metabolic syndrome, musculoskeletal diseases, etc. And given PTSD is associated with elevated levels of inflammation, it's hypothesized that inflammatory responses to PTSD could help explain the link between PTSD and chronic disease. The use of behavioral genomic approaches and other biomarkers offers a means by which to look at how biological mechanisms influence this mental and physical health comorbidity, as well as offers clinical tools for assessing health-related risk and for tracking the efficacy of intervention efforts. So I've used the phrase behavioral genetics, and so it might be helpful to review for a moment why genes matter when it comes to understanding PTSD. There is ample research to suggest that both genes and environment are etiological determinants of PTSD, and that PTSD is moderately heritable, meaning trauma exposure is a necessary but not sufficient criterion for PTSD. And indeed, as the research tells us, genes and environment interact in order to influence the development of PTSD. And that both trauma exposure and PTSD symptoms themselves are associated with genetic factors. There are a lot of different ways to look at how genes influence our mental health and behavior, but for the remainder of my talk today, I'll be talking about DNA methylation specifically. DNA methylation is one step in the process underlying how genetic makeup can affect mental health. So what is DNA methylation exactly? Well, we all have DNA, deoxyribonucleic acid. Comes in the form of a double helix, like you see here on the slide, and it carries with it the instructions for life. So genetic instructions for development, reproduction, functioning, growth, etc. Genes are sections along DNA that are in charge of different functions, like making proteins. So I want you to think about a gene as a door. And if you're thinking about a gene as a door, then we as humans have about 20 to 25,000 doors lined up along our DNA. Now, how these doors function is dependent on a variety of factors that can be influenced by the environment. Certainly included in that is trauma exposure, as well as diet, exercise, a number of environmental factors. This is what we call epigenetics. Now, one epigenetic mechanism that can modify the function of a gene is called DNA methylation. And DNA methylation is a biological process by which a methyl group is added to a DNA molecule, thereby affecting the gene's function and expression. So I'm going to invite you to think about DNA methylation as padlocks that can be added to the doors. And the regions where DNA methylation occurs are called CPG sites. CPG is just an abbreviation used to distinguish the linear sequence of a cytosine pairing with a guanine by a phosphate group, so CPG. And just as a padlock regulates the use of a door, DNA methylation regulates gene expression and gene function. just a quick note that not all DNA methylation markers are functional in this way, and there are researchers continuing to explore the link between DNA methylation and expression. But the research I'll be presenting on today focuses on the padlocks specifically. Okay, so we've reviewed how inflammation might help explain the link between PTSD and chronic disease, and why exploring how epigenetic influences on these associations might be informative. So now let's turn to some of the existing work that's been done in this area that informed the research project I'll be discussing today. In 2016, Lichthart and colleagues published results from an epigenome-wide association study, or EWAS, of C-reactive protein. C-reactive protein, or CRP for short, is a peripheral or blood-based marker of chronic low-grade inflammation, and it's been previously implicated in the pathogenesis of complex diseases. In EWAS, this epigenome-wide association analysis, what happens is epigenetic variants, or indicators of DNA methylation, so think back to those padlocks we just talked about, these epigenetic markers are regressed onto a phenotype of interest in order to detect any statistical associations. So is DNA methylation associated at any location in the genome, across the genome, with CRP, this marker of inflammation? Using EWAS, Lichthart and colleagues found that across 2 large samples of Europeans and African Americans, the strongest predictor of CRP levels was a CpG site located within the absent in melanoma 2, or AIM2 gene. The AIM2 gene serves an essential role in immunity, and it does this via activation of the inflammasome in response to DNA damage caused by pathogens. This mobilization of the inflammasome triggers the production of pro-inflammatory cytokines such as interleukin 18 and interleukin, or IL for short, 1 beta. It also triggers an inflammatory form of cell death called pyroptosis. Thus, this AIM2 inflammasome via pyroptosis aids in removing cells that contain pathogen-related DNA damage, including from the brain. So it really serves to kind of get the bad stuff out, so to speak. This ultimately has a protective effect against disease and neurological dysfunction. It would therefore then make sense that disruptions to this AIM2 pathway have been associated with a variety of diseases, including inflammatory and autoimmune conditions, as well as cancers. AIM2 has also been implicated in neurodevelopment, and research has shown that disruption of this AIM2 pathway is associated with accumulation of DNA damage within the central nervous system, as well as an increased number of neurons in the adult brain, as well as behavioral abnormalities, including pronounced anxiety-like behaviors in preclinical studies. For example, genetic ablation of AIM2, so knocking this gene out altogether in rodents, has been shown to induce anxious behavior in rodents across several behavioral paradigms. So the example you see on the slide here is an open field test, and when the mice kind of stick to the corners rather than exploring the full space, this is a behavioral indicator of increased anxiety. Interestingly, this effect of this AIM2 ablation, so dysfunction to this pathway, appears to be specific to anxiety-like behaviors. Doesn't appear to extend to other behavioral abnormalities like depression, for instance. So given that PTSD is positively associated with inflammation, AIM2 methylation is negatively associated with inflammation. So what maybe a more helpful way to think about this is demethylation at this CpG site is associated with higher inflammation. And disruptions to the AIM2 pathway have been linked to increased anxiety in rodents. We hypothesize that the association between PTSD and inflammation may be mediated by AIM2, methylation. Our group previously tested this hypothesis in a cross-sectional study of 286 veterans with a high prevalence of PTSD, and found that PTSD severity was associated with elevated CRP levels via decreased methylation at this CPG site identified by Ligtart and colleagues. A subsequent study by Young and colleagues, published more recently using a sample of 94 veterans, found that PTSD diagnosis was negatively associated with AIM2 methylation at this CPG site. However, notably, this effect didn't remain significant after adjusting for covariates. Young and colleagues also reported that AIM2 methylation at the site was negatively associated with AIM2 expression and CRP levels. So again, this suggests that lower levels of AIM2 methylation are associated with worse inflammatory outcomes. Our group previously, as well as Young and colleagues here, didn't examine any inflammation markers other than CRP. And to our knowledge, no prior study has examined the associations between AIM2 and CRP or any other inflammatory marker for that matter longitudinally. In this study, we sought to expand upon prior PTSD and animal model research by examining whether the mediating effect of AIM2 methylation extended to other indices of inflammation besides CRP, as well as to indices of neuropathology, and by investigating these effects longitudinally over the course of approximately 2 years. These AIMs were evaluated in a young sample of primarily male post 9/11 US military veterans. The sample was enriched for PTSD as assessed by the clinician administered PTS scale, and processing of the DNA methylation data followed standard procedures previously set forth by the Psychiatric Genomics Consortium. I want to take a brief moment to talk about the technology that was used to assay the peripheral markers of inflammation and neuropathology used in this study, which is called single molecule array, or SIMOA technology. This is state-of-the-art technology that's led to the ability to precisely measure biomarkers at extremely low concentrations. Neuropathology biomarkers measured using the SIMOA technology, including tau, neurofilament light, or NFL, amyloid beta species. Some of these SIMOA measured neuropathology markers have been used to successfully predict Alzheimer's disease among individuals prior to any symptom onset. So I really want to emphasize this point because what it means is that this technology allows for the early detection of biomarkers of health risk before any onset of symptoms. Obviously, this has profound clinical implications for early risk detection and even prevention. Additionally, SIMOA neuropathology and inflammatory markers have been associated with late onset Alzheimer's disease and other neurodegenerative diseases, including multiple sclerosis, Parkinson's disease, and traumatic brain injury. These findings are potentially relevant to PTSD in that PTSD has been shown to increase the risk for development of multiple forms of dementia, including but not limited to Alzheimer's. Furthermore, inflammation is a well-established feature of neurodegeneration. Therefore, evaluating associations between PTSD, AIM2 methylation, and these low-level analytes of inflammation and neuropathology offers a really unique opportunity to look at and hopefully elucidate some of the biological mechanisms underlying the associations between PTSD and downstream neurodegenerative and inflammatory health outcomes. We first examined the bivariate associations between PTSD symptom severity, AIM2 methylation at this locus of interest, Samoa markers, as well as principal ancestry components and estimated white blood cell proportions. The latter two, which were calculated from the genotype and methylation data respectively and included as covariates in all analyses. Because we plan to test our hypotheses using mediation models, we first looked at the bivariate associations between AIM2 methylation at our CPG site of interest and Samoa markers all at time 1, controlling for age, sex, and white blood cell count estimates in order to determine which Samoa markers were associated with AIM2 methylation and were therefore relevant to include in subsequent mediation models. These regression analyses showed that AIM2 methylation was negatively associated with 3 inflammatory markers, IL-6, IL-10, and a tumor necrosis factor, or TNF-alpha. AIM2 was also positively associated with 2 neuropathology markers, the NFL marker I referenced earlier, as well as glial fibrillary acidic protein, or GFAP. I always struggle with that one. These Samoa markers that were significantly associated with AIM2 methylation that you see highlighted here were included in subsequent separate mediation analyses. One focused on the inflammatory markers and the second on the neuropathology markers. Here you see the results for the cross-sectional mediation analysis examining the direct and indirect associations between PTSD, AIM2 methylation, and the 3 inflammatory markers. And what is noted here on the slide and subsequent slides to come are the standardized betas and standard errors, just to note. The model showed significant direct associations between PTSD symptom severity and AIM2 methylation, as well as AIM2 methylation in all 3 inflammation markers. There were significant indirect effects of PTSD on all 3 inflammation markers through AIM2 methylation, all of which survived correction for multiple testing. This mediating effect was replicated longitudinally for one of the markers, such that AIM2 methylation at time 1 significantly mediated the relationship between PTSD symptoms at time 1 and IL-10 at time 2, controlling for relevant covariance as well as the autoregressive effect of IL-10 at time 1. Essentially, what this means is that the mediating effect of AIM2 methylation explained variance beyond what would be expected or accounted for by baseline levels of IL-10. There was no evidence for any longitudinal indirect effects on either IL-6 or TNF-alpha at time 2. We conducted a follow-up longitudinal analysis to test for bidirectional associations and didn't find any evidence for the reverse direction of effects over time. We also wanted to test whether the mediating effect of AIM2 methylation on the relationship between PTSD symptom severity and CRP levels that this research group, I wasn't part of that project, but the research group that I'm affiliated with now previously reported on in their 2018 paper. We wanted to see if that cross-sectional effect replicated longitudinally. And we did not find evidence of any longitudinal replication in the cohort that we had and the data that we had. Turning our attention to the neurology markers, this slide shows the results for the cross-sectional mediation analysis examining direct and indirect associations between PTSD symptom severity, AIM2 methylation, and NFL and GFAP all at time 1. And the model showed a significant negative association again between PTSD and AIM2 methylation. As well as a positive association between AIM2 methylation and NFL, but not GFAP. There was a significant indirect association between PTSD symptoms and NFL through AIM2 methylation, which did survive correction for multiple testing. And the indirect effect of PTSD on GFAP via AIM2 methylation was not significant. There was no significant indirect effect of PTSD symptoms in the follow-up longitudinal model predicting NFL at time 2. And the examination of the reverse direction of effects over time, there was no evidence that time 1 NFL mediated the association between AIM2 methylation at time 1 and PTSD symptom severity at time 2. Again, controlling for the autoregressive effect of PTSD severity. Okay, so let's review, just to review here, the results suggested the importance of AIM2 as a biomarker of multiple peripheral indices of both inflammation and neuropathology that have using markers that have been previously associated with adverse health outcomes like Alzheimer's, multiple sclerosis, Parkinson's, and TBI. However, the direction of these associations differed for the inflammation and neuropathology markers. Specifically, greater levels of PTSD symptom severity were associated with higher levels of inflammation via decreased AIM2 methylation in the cross-sectional analyses. This is consistent with the 2 prior studies that I referenced earlier implicating AIM2 in PTSD-related inflammation. In addition to these cross-sectional indirect effects, results also showed that an indirect longitudinal effect of PTSD symptoms on IL-10 was statistically significant at a later time point via AIM2 methylation. IL-10 is typically considered an anti-inflammatory cytokine, whereas the other 2 markers we looked at, IL-6 and TNF-alpha, are pro-inflammatory. Despite this, research suggests that all 3 of these cytokines are activated or increase in the acute phase and chronic inflammation, kind of following a stressor. So for example, IL-10, this anti-inflammatory cytokine, might increase in the face of pro-inflammatory molecules to act as this mechanism to generate homeostasis or kind of temper inflammation. So you might also see that increase along with pro-inflammation. And this is consistent with our findings that showed positive correlations between IL-10, IL-6, and TNF-alpha, as well as the consistent direction of effect with regard to the cross-sectional associations between PTSD, AIM2, and all 3 markers. Interestingly, however, whereas the cross-sectional mediation analysis suggested PTSD severity increased levels of all 3 markers via decreased AIM2 methylation, the longitudinal model showed that PTSD severity was associated with decreased levels of IL-10 over time via AIM2 methylation, again, beyond what would be expected based off of baseline levels of IL-10. This suggests that increases in both pro and anti-inflammatory responses may co-occur at any given time point in association with PTSD and AIM2 methylation, but that over time, PTSD symptom severity is associated with decreased anti-inflammatory responses via decreased AIM2 methylation. This suggests kind of a reduced capacity to mount an anti-inflammatory response over time in the presence of higher levels of PTSD. We've just reviewed how greater PTSD symptom severity was associated with increased inflammation via AIM2. Conversely, greater levels of PTSD symptom severity were associated with decreased levels of neuropathology via decreased AIM2 methylation, suggesting that AIM2 methylation may have some sort of a neuroprotective role in decreasing the effect of PTSD on neurodegeneration. Although this was contrary to our original study hypothesis, which was based off of this abundance of literature implicating inflammation in neurodegeneration, This finding is consistent with a body of research suggesting that AIM2 limits neurological dysfunction, again, by way of inflammasome-induced death of damaged cells, of damaged DNA rather. So in other words, one of the functions of increased AIM2 related inflammation may be to leverage these inflammatory responses in order to induce cell death among damaged cells that would otherwise promote neurodegeneration. Therefore, PTSD might serve to activate the AIM2 inflammasome via decreased AIM2 methylation and increased AIM2 expression, thereby activating pro-inflammatory cytokines that you see all the way over on the right in this figure on the left, that would then defend against pathogens in the brain. There are a number of clinically meaningful implications to these findings. AIM2 methylation was associated with markers of both inflammation and neuropathology in this sample, even at very low levels, suggesting that this may serve as a useful screener or biomarker for multiple adverse outcomes associated with PTSD. AIM2 methylation might also be useful for tracking responses to early prevention and intervention techniques. So for instance, exercise or diet interventions aimed at alleviating inflammation. AIM2 could be a useful screening tool or tracker to test those or other psychological or pharmacological interventions to address PTSD symptoms. Further modulating AIM2 methylation through therapeutic drug treatments, for instance, might yield changes in inflammatory and neuropathology markers, which could influence risk for negative health outcomes. The young age of our cohort really demonstrates the importance of using Simoa technology to detect very, very low, presumably initial, prior, could be years prior to any symptom onset, and yet you're able to find within the blood these very low, presumably initial levels of markers of later downstream serious health outcomes and adverse effects. So given that these adverse processes might begin at a very young age, this association between AIM2 and Simoa markers may be important for intervening at this early stage when disease trajectories may be reversible. One caveat to this that's important to note is that we were unable to determine if AIM2 methylation is a biomarker for inflammation and neuropathology risk, or part of the mechanistic pathway for such pathology. Certainly, future research is needed to address this question. For the sake of time, I won't spend too much time on study limitations. However, there were some. First, the sample was relatively homogeneous with respect to sex, ancestry, trauma type. Also, so that limits generalizability to other populations. Second, the power to detect possible longitudinal effects across the markers might have been limited due to sample size. It might also have been limited due to the restricted time interval between assessments, which in the cohort that in the data I presented on here was about 2 years. So it's possible that might be too short to observe the cumulative burden of chronic PTSD symptoms on these peripheral analytes over time. Certainly an interesting area for future research to expand this time frame between assessments, as well as increase sample size, all of which might lend a better understanding to the longitudinal effects. Additionally, the analyses were limited to data derived from samples in the blood. It's possible some of these effects could be tissue specific, which we weren't able to look at, or certainly look at in the brain, probably has some real relevance to this research question. Broadly, the findings of this study suggest that AIM2 DNA methylation may be a useful and cost-effective clinical target that is able to singularly capture multiple biological markers associated with trauma-related risk and resilience towards inflammatory and neuropathological conditions. Dr. Wolf, who is my mentor and is truly fantastic, and I've had a wonderful 2 years of fellowship with her, this is my shout out here on the disclosures and funding slide, but she is senior author on this paper, owns some stock in Illumina, but other than that, there are no conflicts of interest to disclose. And I'd like to wrap up by again expressing my appreciation for having received the Donald F. Klein ECI Award. Thanks to this award, I had the pleasure of attending my first ADAA conference, which was a very refreshing blend of both basic and applied topics. I really enjoyed my experience at that conference, and also found that it was a really, really helpful network opportunity. There are additional awards in addition to the Donald F. Klein Early Career Awards that provide young and early career investigators opportunities to have their work recognized and acknowledged. And through that, I was able to meet a lot of really interesting young investigators doing really interesting work, and am excited and hopeful about the collaborations that just that experience will have fostered. So I'm very grateful for the opportunity to share my work with others through this, through receiving this award, and to have also expanded my academic connections. So thank you very, very much. I'm very honored to have received it. With that, I thank you all so much for your attention. If the pandemic has taught me anything, it's that people show up to virtual talks or meetings, what have you, for the animals. So here's my fur baby, Wilson. And I'll leave you thus with Wilson, as well as my contact information here on the slide. Please feel free to contact me if you have any questions or thoughts about anything I've presented today. Thank you so much.